Next concept, that is structure of bacterial cell, that is under the cell organization. So what is the bacterial cell structure? So now let's move on to it. So bacterial cell, we bacterial cell is composed of mainly three components, that is cytoplasm, that is inside the bacterial cell. Here you can see this is the inside of bacterial cell, the whole is a cytoplasm. And next, that is cell envelope that covered the bacterial cytoplasm, and that another one is a external structure, that is present outer side of the cell envelope, like this one, this flagella or pili structure. These are the external structure of bacteria. So bacterial structure is composed of three main components, that is cytoplasm, cell envelope, and external structure. So within a cytoplasm, main two types of things are there, that is cytosol, that is the liquid portion. Here the liquid, all these are the liquid portion known as cytosol, and the solid portion, solid things which are present, that is the internal structure. And within the internal structure, one thing is nucleoid, where DNA is present. This is the nucleoid where DNA is present, that is the genetic material. And another one is ribosome. Here these are the ribosomes, these are the ribosomes where protein synthesis occur. And another thing which is present in cytoplasm, that is plasmid. This is the plasmid, this is the plasmid, this is the plasmid, that is a extra chromosomal DNA structure. It is also contain DNA, but it is not the genetic material. Okay, and in our another video we will explain this plasmid structure and function differently, okay, in detail. And another one, that is inclusion bodies. So inclusion bodies, this is the inclusion bodies where bacteria store some foods like polysaccharide, phosphates, many types of foods are there for their future. Okay, and the cytoskeleton means the it provide this it control the shape of the bacterial cell. Okay, regulate the shape of the bacterial cell, okay, or protect it. So this is the whole cytoplasm structure. Okay, and which one covered this cytoplasm structure is known as the cell envelope. And in cell envelope, that is present the cell membrane and cell wall. So mainly the cytoplasm is covered by the cell membrane. So what is cell membrane? So cell membrane covered this cover, this portion, this cover is the cell membrane structure. Okay, and outside of this cell membrane, the component is the cell wall, that is that white portion, or outside this white portion, this is the cell wall. Okay, this is the cell wall. Okay, and what is the external structure? External structure, within an external structure, we can say the first one is the flagella. So this is the flagella which help the cell for movement or motility. Okay, and the pili structure, here this one is the pili structure. Pili helps the bacteria for bacterial mating, like bacterial DNA or bacterial nucleoid or bacterial genetic material can be transferred from one bacterium to another bacterium. Suppose this is a bacteria and this one is another bacteria. So for their bacterial mating, the in some bacteria, the genetic material, that is DNA, can be transferred from one bacterium through pili to another bacterium. So this process can be done by pili, and also pili helps in attachment to another bacteria or another surfaces. Okay, so this is the role of pili. And another thing that is contained in the external structure, that is glycocalyx. Glycocalyx are the sticky components which are present outside of the cell wall, that is known as the pili structure, glycocalyx. And within a glycocalyx, the glycocalyx can be a slime layer or capsule. If the glycocalyx is contained in very tightly, then it is called the capsule, and if contained very loosely, then it is called slime layer. Okay, so glycocalyx, main role of glycocalyx is to stick to a surface. So it helps the bacteria to attach any surfaces. Okay, so these are the all structure, and now we will see the bacterial cell in detail. So these are the pilies. Here we can see these are the pilies which help the bacteria for transfer of genetic material from one bacterial cell to another bacterial cell. Okay, and next, that is the glycocalyx. This sky color thing is the glycocalyx, that is the sticky portion for attachment. And next is the cell wall. This is the cell wall which gives a shape, and it protect the bacterial cell, or it act as a protection layer. Okay, next, inside the cell wall, the present component is the cell membrane. This is the cell membrane. So outside of the cytoplasm, the cell membrane is present, which is a lipid bilayer, which is a semi-permeable membrane. By this semi-permeable membrane, selective components can go outside of the cell or go inside into the cell. Okay, so selective components can be transferred from that. So that's why it is called semi-permeable membrane. Okay, so this is a cell membrane, and then we can see in a cytoplasm nucleoid is present, which is with DNA. So here the DNA or genetic material is present, and next component is inclusion bodies. So here the storage components are present, or polysaccharide, many things are stored in this components. Okay, and next is plasmid. So this is the plasmid, that is a extra chromosomal DNA material. Here some genes are present which help the bacteria to resistant from external external factors. Okay, or to be virulent. Okay, so it helps the bacteria virulence. Okay, so another thing, that is the flagella. So flagella helps the bacteria for their movement. Okay, so now move on to the more detailed structure. So here we can see, so first thing is the glycocalyx. So what is glycocalyx? We will see here a coating or layer of molecules external to the cell wall, and it is present in the cell wall. Outside of the cell wall, that is a coating or layer, it is a sticky layer, and it serves protective, adhesive and receptor functions, and it may fit tightly or be very loose and diffuse. So if it is present in tight form, that is known as a capsule, and if it is present in loose form, that is known as slime layer. Okay, and then that is a bacterial chromosome or nucleoid. So here this is the component. So this is the bacterial nucleoid structure. So it is a composed of condensed DNA molecules. Okay, here the DNA molecules are present, that is the genetic material, and DNA directs all genetic and heredity of the cell and codes for all proteins. Okay, so next one is the pili. So this one is the pili, an elongated hollow appendage used in transfer of DNA to other cell. This nuclear or genetic material, or this plasmid, can be transferred from from one bacterial cell to another bacterial cell by this pillar structure. The process is known as conjugation. Okay, so next is plasmid. So what is plasmid? Plasmid is a double stranded DNA circle containing extra gene. Here also some genes are present which protect the bacteria or give some extra advantages to the bacteria. Next one is the ribosome. Teeny particles composed of protein and RNA that are the sites of protein synthesis. So this is the place where protein synthesis is performed. Okay. So next one is actin cytoskeleton. Long fibers of proteins that encircle the cell just inside the cell membrane and contribute to the shape of the cell. So it gives the shape, a specific shape of the bacterial cell. Okay. So next one is the flagellum or flagella. So it is a specialized appendage attached to the cell by a basal body that holds a long rotating filament, and the movement pushes the cell forward and provide motility. So it basically gives the bacterial cell its motility. It helps in motility. Okay. Next one is the cytoplasm. So it is the water based solution filling the entire cell. So this is the cytoplasm. And endospore, so what is endospore? It is a dormant body formed within some bacteria that allow for their survival in adverse condition. So in adverse condition, some bacteria, suppose some bacteria are placed in a 75 degree Celsius environment or in a very dry environment. So in that time, in that adverse condition, for their survival, bacteria go to their dormant state. Okay, that means they do not divide or do not reproduct. So that's time, that is called the dormant state. So that that portion is known as the endospore structure of bacteria. So when after some times, when the bacteria get a favorable condition, that bacteria from their dormant state come to their favorable vegetative body or normal bacterial cell. Okay, so this is a survival strategy of the bacteria. Okay, so next is the outer membrane. So extra membrane, similar to cell membrane but also containing lipopolysaccharides. So outer membrane basically present outside of the cell wall. It is mainly present in the gram negative bacteria. Okay, so we will all discuss about the gram positive, gram negative bacteria in our next videos when we discuss about the cell wall of bacteria. Okay, so outer membrane and contain flow of materials and portions of it and are toxic to mammals when released. Okay, so what is cell membrane or plasma membrane? So plasma membrane, all we discussed here, that is a thin sheet of lipid and protein that surrounds the cytoplasm and controls the flow of materials into and out of the cell pool. So the plasma membrane basically composed of lipid and protein, which is known as a lipid bilayer, and it is not a solid structure. Okay, it is a fluid structure and it is a semi-permeable. By it, many selective materials can go into or go out of the cell. Okay, and the next thing is the cell wall. So it is a semi-rigid structure that that provide structural support and shape for the cell. Okay, and last thing, that is, oh sorry, another thing, that is inclusion bodies, that is a stored, it is a storage compound where bacteria store their nutrients such as like fat, phosphate or glycogen deposited here. Okay, and last thing is fimbriae. These are the fimbriae structure. So fimbriae, that is a fine hair like bristle extending from the cell surface that help in addition to other cell and surfaces. So it basically helps to adhere to the other another cells or to another surfaces. So it help in addition. Okay, so this is all about the function of cell structure components. Okay, so more specifically here we can say that is plasma membrane structure. So what is their function? So the function of the plasma membrane is to selectively permeable barrier. So it can selectively permeable for many components. It can only support selective components to inside or to outside of the cell, and it can act as a mechanical boundary or mechanical barrier of the cell and nutrient and waste transport and location of many metabolic processes like respiration and photosynthesis, and detection of environmental cues for chemotaxis. Okay, so next one is the gas vacuole. So what is the function of gas vacuole? So it provides the buoyancy for floating in aquatic environment. For in a aquatic environment, for floating of the bacteria, it helps in floating of the bacteria. Okay, and next one is the ribosome. So the main function of the ribosome is to helps in protein synthesis. It gives the place for protein synthesis. And next one is the inclusion bodies. So that is the storage of carbon, phosphate and other substances. And nucleoid means localization of genetic material, that is DNA. And next one is the periplasmic space. So what is periplasmic space? Periplasmic space means, here you can see this one is the cell wall and this one is the cell membrane. Okay, so in between the space, this space is known as the periplasmic space. This is the periplasmic space. Okay, so what is the role of periplasmic cells? So it contains hydrolytic enzymes and binding proteins for nutrient processing and uptake. So if we get a complex nutrient, then in the periplasmic space, many hydrolytic enzymes are there which break down those complex components and uptake the more easy or more soluble or more simple compound inside the cell. Okay, so next one is the cell wall. So what is the role of cell wall? It provides shape and protection from osmotic stress. And next one is the capsule and slime layer. So they are the glycocalyx. So what is the role of them? So resistance to phagocytosis and adherence to surfaces. So they helps in adhere to different surfaces because there's a sticky layer. Okay, so next one is the fimbriae and pili. So their role is basically attachment to surfaces. That is the role of the fimbriae. And bacterial mating. In bacterial mating helps, it helps, pili helps in bacterial mating for transfer of the genetic material from one cell to another. Okay, so next one is the flagella. So their role is in motility. And last one is the endospore. So their role is to survival under harsh environmental condition. Okay, so these are the role of bacterial structure and their function. Next is the size